Hello.
Yes, yes.
Hello. Am I audible?
Sir.
Okay. So, okay. I think you people are enjoying this training session. And I am Dr. Subhradip Bhattacharjee. On behalf of Growing Seed Organization, welcomes you once again to this training session. Today, the lecture will once again be delivered by Dr. Rajat Nath, Research Scientist, Department of Life Science and Bioinformatics, Assam University. So yesterday he had discussed about computer-aided drug designing, and today also he will continue his session. So without much delay, I am transferring this session to Dr. Rajat Nath. Dr. Rajat Nath, we heartily welcome you to this session. Thank you.
Thank you, Subhradip, and good evening, everyone. So, yesterday in our yesterday's class, we have discussed about the retrieval of the ligand structure from the database, and we have drawn the structure if it is not available in the databases. We have seen how to get the chemical properties using the chem sketch and chem draw. Then we have seen the toxicology prediction of the ligand. So, have you anyone tried at home and facing any problem regarding this? Okay. So, anyone, if you required any part of the yesterday class have to revise, you may say then I will revise that yesterday's class, then to start today's session, if anyone needed. Otherwise, I will start today's class. Okay. So, very rapidly, I will tell what we have done yesterday. My screen is visible now.
Sir.
Okay. So, first of all, we retrieved the ligand structure from the database. The database was PubChem. PUBCHEM. PubChem was the database. We written the name of our compound here, like anything, like yesterday we tried using aspirin. So today I'm searching another compound known as quercetin. I'm opening the ChemDraw. draw a structure. Just a simple structure I am drawing here, like that. This is a benzene. One. Let's think this is one a simple structure I have drawn. So I will select this structure and I will open the chem sketch. So chem sketch. Then I will paste the structure. This is my structure. To get the smile notation, I will go to the tools, go to the generate, and click on smile notation. The smile will be pasted in the workspace. If I want to draw this structure from this smile, what I can do, I will copy this smile, go to tools, go to generate, and structure small smile. So as I already selected this smile notation, it came automatically. Otherwise, you can just paste the smile and click OK. This structure will be come in this the workspace. If we need the name of the structure, we will just select the structure, go to tools, generate a name for the structure, and the name of the structure will be shown here. So if we want to convert this smile notation to SDF format, just copy this structure smile, go to open Babel, in left hand side select the input format. Here already selected a smile. I will pasting this smile here, giving a space and giving one name. I don't know the name of this structure. I'm writing just compound one. And left hand side select the output format. I am selecting output format SDF, and below the output format I will select the path where the structure will be saved and give one name. Then click generate 3D coordinates and then convert. It will convert it in SDF format. In desktop, you can see this component has been converted from smile format to SDF format. Okay, so that this was the process how we draw the structure and how we predict the name and predict the smile format of the structure, and from the smile how we converted the structure to SDF format. Now this is the PubChem database. We search a compound named quercetin, and if we click on this quercetin, we will get the canonical smile here. This is the canonical smile for the same. If we select this canonical smile, copy it, go to Chem Sketch, go to Tools, Generate, Structure from Smile, and click this. The structure will be generated here. Okay, so this is the process how we can get the canonical smile from the PubChem server. Then there is IUPAC name of the structure. So we will just select this IUPAC name, Ctrl+C to copy it, go to ChemDraw, go to structure, then convert name to structure, give the name, click OK. The structure will be there. So from the IUPAC name, we can predict the structure. Okay. To download the structure, just go to the download option, select the 3D conformer and save as SDF. It will be downloaded. So I am writing its name as quercetin and saving it. when save, after saving, I will just right click it, open with Notepad. Then just change the first line as its name without any space. And Control S and close the Notepad. Then this ligand will be downloaded and its name will be changed from PubChem ID to its compound name. Then we have studied the toxicity of the compound. For that, we have used TOX2. Sorry, TOX2. This is the TOX2 server. Then we go to the toxicity. Then paste the canonical smile. Just paste the canonical smile. Click the smile. Then select the organ toxicity. Just check all the box. Whatever you are required. Then click start tox prediction. It will give the toxicity result. In prediction, it is showing the calculating. So this is the toxicity result of this compound. Then for the target prediction, we used Swiss target prediction. We have pasted our smile here. Then clicked on predict target. And the target was predicted like that. In yesterday's compound, only 15 to 16 compounds are showing prediction. But today's compound, see, in all the 100 targets are showing some kind of prediction that having the interaction with the targets. And most of the compounds having the 100% chances of getting the interaction with the target compound. So depending upon the types of the compound, the targets and the probability will change. So that much we seen yesterday's class. So today we will learn about the ADME profile. So what is ADME? So ADME is the absorption, distribution, metabolism, and excretion, as I have told yesterday's class. So actually this word is 5-letter word, that is ADMET, A-D-M-E-T. T stands for toxicity, which we have already studied yesterday. So today we will see the first 4 letter, that is A-D-M-E. A means absorption, D means distribution, M means metabolism, and E means excretion. To get the result of ADME, we have to go in one website known as Swiss ADME. Just type Swiss ADME and open the first link. This server also created and maintained by the Swiss Institute of Bioinformatics. Yesterday we have used Swiss target prediction. This also created and maintained by the Swiss Institute of Bioinformatics. So, if you want to know more about this ADME profiling and the meaning of the score, how these scores are calculated, and what is the meaning of this score, you can simply select those three research paper they have mentioned here in red color. Just open these three research paper. One is published in Scientific Reports. It is Swiss ADME, a free web tool to evaluate pharmacokinetics, drug likeness, and medicinal chemistry, friendliness of small molecule. Another publication is ILOG-P, a simple, robust, and efficient description of an octanol water partition coefficient for drug design using a GBSA approach. And third paper is a boiled egg to predict gastrointestinal absorption and brain penetration of small molecules. If you study in this three paper, you can easily analyze the result retrieved from this website, and you can easily understand the meaning of this result. What is the meaning of the various parameters? What is the meaning of the scores derived from this server, and what score should be? What is the range of the score should to be a good drug candidate? Okay, so I will recommend, please download those three papers and go through it once, so that you can understand this more accurately. So, to predict the ADME profile of any compound, we will again visit the same website, which is the database for small molecules or ligand, and will search any ligand name of your interest. Like I am searching here one compound known as gallic acid. Okay, so this is the gallic acid. So I need its mile format, because in this server they have written the enter a list of smile here. So in this profile, you can analyze the ADME profile of more than one compound at a time. As we have seen in Protox, we can analyze the toxicity of a ligand, single ligand at a time. In our Swiss target prediction, we have seen we can study the target prediction for single compound at a time. But in case of this Swiss ADME studies, we can paste here more than one compound at a time. Maybe you can enter 300, 400, 500 smiles here at a time. So how we can enter the smiles in this portal? So first of all, you just need to open one notepad. I am writing this name as compounds. Just open one notepad. This is the notepad I have created. So this is on a notepad. So what can I do? I will go to the PubChem server and search on compound known gallic acid. So this is the gallic acid, and what I will do, I will just copy the canonical smile. This is the canonical smile of gallic acid, and I will paste it in the notepad. Then I will give one space and will write the name of this compound. This is gallic, but I can't give any space here. So I will use one underscore before writing acid. Okay. Then click enter. Then go to the PubChem again. Search another compound like I'm typing quercetin. Go to the quercetin. Select the canonical smile. copy the smile, paste in the notepad, give one space, and write down the name where setting. Then click enter. Go to PubChem again. Search another name like aspirin. Click aspirin. Scroll down below. Select the canonical smile. Control C and paste here by clicking Control B, space aspirin. Okay, so like that you can make a list of compounds of your as your experiment design needed. So you can put 200, 300, 400, how many compounds you want, you can put here. So simply just select all by clicking Control A, copy all with clicking Control C, or right click copy, go to admit and paste the list here. Okay, just paste your smile list here. Then click on run. So it will take few minutes to generate the data. See what happened. As I mentioned their name, so the result came here with its name. See, first compound is gallic acid, it's named as gallic acid. If you paste only smile, there will be written as molecule 1 and molecule 2, molecule 3. But if you put the name here, then the result will come with its name. So this is the first result for the first compound, which is gallic acid. Scroll down, this is the second compound, quercetin, the result for the quercetin. Scroll down below, this is the aspirin, and it is the result for aspirin. So you can put as much as compound you need, and you will get the result. And you can download the result at a time by clicking retrieve data. In right hand side there is an option, writers retrieve data. Just click on CSV file, CSV icon showing here. Click this CSV icon and give a name and click save. It will be saved as a CSV file. So this is the file I have saved. So you can see the result of the ADME is retrieved in a Excel file. So in first column there is the name of the compounds. In second column there is the canonical smile. Third column formula, molecular weight. There is so many data generated from this ADME analysis. So what is the meaning of the different parameters? Okay, so there is the parameter known as heavy atom, aromatic heavy atom, fraction, rotatable bond, hydrogen bond acceptor, hydrogen bond donor. There is so many parameters available in this Excel file. So what is the meaning of these parameters? And there is some code. So what is the meaning of this code? What is the range of this code should be there to become a drug as a good, having a good ADME profile. So this you can easily understand if you go through this three literature. Okay, if you go through this literature, you can get the idea about what is the meaning of this parameter and what is the ranges required for these parameters. So the result we have get in the Excel file is also written in this window. Okay, so simply I am telling you, this is the formula weight. This is the formula of this compound, that is gallic acid, and this is the formula of gallic acid. Simpler formula is C7H6O5. Then it comes molecular weight. So why molecular weight is important? Because I have already told in yesterday's class that in Lipinski rule of 5, molecular weight should be less than or equal to 500. So molecular weight is a very important parameter for a drug candidate or become a drug-like compound. So here in physicochemical properties, this molecular weight is written. Number of heavy atoms is 12. Number of aromatic heavy atoms is 6. In fraction CsP3, number of triple bond 1, number of hydrogen bond acceptor. So see, and number of hydrogen bond acceptor and number of hydrogen donor is most important, because I have already mentioned that according to Lipinski rule of 5, there should be hydrogen bond acceptor 5 and hydrogen bond donor 10, less than 10. Then come molar reflectivity and TSPA. So what is TSPA? If you click on this question mark written here, you can get the full form of every parameter. So here TSPA stands for topological polar surface area. So what is the topological surface area? If you want to know about this, you just go through the literature mentioned here. Okay. So by that you can get the idea of all the parameters written here, because in all parameters there is a question mark and given in the citation is given. You can go and read. Now I will discuss the parameters which is very important in requirements in computer-aided drug designing. Okay, so I will discuss about those parameters, what is the meaning of these parameters and what should be the score of those parameters. So first one is molecular weight. As I already mentioned, it is one of the required parameters for drug designing. Second one is number of hydrogen bond acceptor, number of hydrogen bond donor, which I have already mentioned in yesterday's class. Then this is the water solubility. So there are 3 algorithms used to test the compound, is it soluble or not. So these are the 3 different algorithms used by the software. One is log S by ESEOL. This method is known as topological methods implemented from Delaney JS 2004. This is the one algorithm used by the one scientist. This is another method using by another scientist, and this is the another method using by another scientist. So all the methods are used to identify or predict the water solubility status of this compound, but they have used 3 different algorithms given by 3 different scientists. Okay, so according to log S, that is by ESOL, this compound is showing very soluble. That means it is very much soluble in water. According to algorithm given by the Ali, this compound is also showing the soluble. According to the algorithm given by the Silicos IT, the compound is also showing the water soluble. So from this result, we can say that our compound is water soluble. Now, according to your requirement or drug delivery system or target of the target situated in your biological system, you can know that is your compound should be water soluble or not. According to your requirement, the compound should water soluble or not. According to your research, you can get that your compound is water soluble, and if your compound is requirement is water soluble, then you can go for it because it's meeting your criteria. But if your compound is required, it should not be water soluble, it should only be soluble in fats, then you can reject this compound according to your requirements. Then it's come GI absorption. GI absorption means gastrointestinal absorption. So GI absorption is high means it's a very good parameter for become a drug, because if the compound or ligand is not absorbed by our GI, that means gastrointestine, then it can't be distributed in our body or in our target area. So it should be high. So our compound is showing GI absorption as high. Then come BBB permanent. So what is BBB? BBB is blood-brain barrier. Okay. So If BBB, that means blood-brain barrier permeability, is showing no, that means your compound will not permeable to blood-brain barrier. And if it is showing yes, that means this compound is able to permeate the blood-brain barrier. But now you can say that what should be the result to become a good drug? It should is it required to cross the blood-brain barrier, or it not? It should not be permeable to blood-brain barrier. So the simple answer is, let us consider you are working with a neurodegenerative disease. Okay, you are working with a neurodegenerative disease and working on brain. So if you target something which should cross blood-brain barrier to get its target, then if it is not permeable to blood-brain barrier, your compound will not get distributed to your target and have no use of your compound. So if you are working with any neurotransmitter, you are working with any disease like Alzheimer's disease, you are working with like any Parkinson's disease or any other neurodegenerative disease, then it should be blood-brain barrier permeable. Otherwise, it can't go to the our CNS or central nervous system. But if you are working in other disease, like you are working on hepatocellular carcinoma, you are working on diabetes, you are working on heart disease, cardiovascular disease, you are working on nephrodegeneration. So it should not be permeable to blood-brain barrier, because it will unnecessarily permeable to and target to central nervous system and may affect some unavoidable unnecessary action will be shown after interacting with the central nervous system. So it will be better if you are working other than any disease of neurodegenerative disease, then it is better that it is not permeable to blood-brain barrier. Okay. Then it is come PGP substrate. PGP substrate, it stands for P glycoprotein substrate. So what is P glycoprotein substrate? P glycoprotein substrate is yes or no. That means if P glycoprotein substrate is showing yes, that means it can efflux out your compound from the blood-brain barrier. If your compound is permeable to blood-brain barrier, and if the compound is showing PGP substrate true, then PGP substrate will efflux out your compound or ligand from the blood-brain barrier. And if the PGP substrate is no, that means it can't efflux out your compound from the blood-brain barrier. So in case of, again, if it is you are working other than the neurodegenerative disease, then it should be PGP substrate yes, if the compound is blood-brain barrier permeable. So what will happen in that case? If your compound or ligand cross the blood-brain barrier, PGP substrate will efflux out from the blood-brain barrier and it will not be interacted with central nervous system. But if you are working with neurodegenerative diseases and you targeted this compound to permeable to blood-brain barrier, then if PGP substrate is true or it is yes, then your compound will be efflux out and it can go to the CNS and target your target the protein you have designed to inhibit. Okay. So in case of neurodegenerative disease, PGP substrate should be no, or in case of other disease rather than the neurodegenerative disease, this PGP substrate should be yes. Then there is some other inhibitor like CYPA12, CYPA2C, 119. These are the various substrate inhibition. You can go and search the meaning of this type of compounds, what is the necessity of these inhibitors. So we are not considering that there. Then comes drug likeness. So in drug likeness, there are one that is Lipinski. So as I mentioned, what is Lipinski? That is, if when I click on the question mark, it will see that molecular weight should be equal to less than 500. M log P should be less than or equal to 4.15. It is actually 5. Number of hydrogen acceptor 10, number of hydrogen donor 5. So this is the rule of Lipinski, and it is showing that there is no violation of Lipinski rule of 5. There is showing 0 violation. That means our compound is following the rule of Lipinski rule of 5. There is another rule of Lipinski rule, that is another drug likeness rule known as Gauche. So what it says, the Gauche says that the molecular weight should be less than or equal to 160, greater than or equal to 160 and less than or equal to 480. The W log P should be less than or equal to 5.6 or greater than or equal to 0.4. So these are the different parameter or criteria given by the different scientists, and your compound is showing that Gauche, it is violating 2 parameters given by the Gauche. One is MR is higher than 40, another is atoms is higher than 20. There is another drug likeness rules given by Weber, and your compound is showing yes, that means it is not violating its rule. There is another rules given by the Egan, and it is also your compound is showing it is not violated by your compound. Then another rules given by Muji, that is known as Bayer filter. According to these rules, it is showing that your compound is violating its rule regulation, because it is showing that the molecular weight should be greater than or equal to 200 or less than or equal to 600. But in case of your compound, the molecular weight is 170. So in that case, your compound is violating the rule of Muji. So as in our case, we use Lipinski rule of 5 to analyze our drug likeness properties. So you can consider this Lipinski rule of 5. Other than this, you can give the result of other rules in your manuscript to get the mood result. Then came the bioavailability score. That means what is the meaning of this bioavailability score? After getting the or injected the compound in the biological system, what percent it will be remain to act to show the bioactivity by the compound. So 0.56 is very good core score as a bioavailability score. If you consider in percent, it is 56% of your ligand will be interact with the target and show its bioactivity. Then there is come another medicinal chemistry parameters. One is pains. Pain is stands for pain as a interface structure, and another one is brain structure. So these are the medicinal chemistry properties. It is not required. So you can select few of these parameters for ADME profile. As I mentioned, that is molecular weight, number of hydrogen bond acceptor, number of hydrogen donor, the water solubility. You can add the pharmacokinetics result. You can add the drug likeness result. You can add bioavailability score. Those scores are important for your publications. Now I will click on the show boil egg. So when I click on the show boil egg, what will see? One egg-like structure. This is the yolk of an egg, and this is the white part of your egg. And there is some dot is there. So what is the meaning of this boiled egg? In left-hand side, you can see that show molecule name. So if I click here, you can get the molecule name here. That means aspirin is situated in the yolk part, and gallic acid and quercetin is situated in the white part of the egg. So what is the meaning of this? In the legend, you can see yellow part means BBB, that means blood-brain barrier. White means HIA, that means human intestinal absorption. Blue is PGP plus, that is PGP positive, and red means PGP minus, that means PGP negative. So PGP, what is PGP? I have already said P glycoprotein. So PGP means P glycoprotein. So from this boiled egg, what can I get? That there is three compound. One is aspirin, which is crossing the blood-brain barrier. That's why it is in the yolk part of the boiled egg. Okay, so I can see that aspirin is blood permeable to the blood-brain barrier, but quercetin and gallic acid is not permeable to blood-brain barrier, but having the good gastrointestinal absorption. If this compounds is situated in the uh, outside of this egg, if it is situated like that area, that means it is not good, it is not having the good absorption in the gastrointestinal tract. That means the gastrointestinal tract absorbance is very low of this compound. So in this case, our compound is showing good gastrointestinal absorption. That red color means this compound is PGP negative. That means if it enter to the blood-brain barrier, it will not be efflux out. Okay. So if quercetin is enter in the blood-brain barrier, it will not be efflux out because it is PGP negative. So as our compound is not permeable to blood-brain barrier, so I don't have to see is this compound PGP positive or negative, because if this compound is not going to the blood-brain barrier, then what is the meaning of seeing that is it will be efflux out or not? It is not matter of concern because it is not going to the permeable blood-brain barrier. Then came the aspirin. In case of aspirin, it is showing again PGP negative. That means this aspirin can't be efflux out from the blood-brain barrier. So if aspirin is using a disease rather than the related to neuro, then it will be go to the central nervous system and interact with the central nervous system because it can be efflux out from the blood-brain barrier. But if it is acting with any neurodegenerative diseases, then aspirin can be done because it will permeable to blood-brain barrier and it will not be efflux out by the PGP glycoprotein. Okay, so this was the boiled egg structure. So you can just take a screenshot of this structure. This is the screenshot I am taking like that. Save this structure as I am saving this structure as compounds. So this is the boiled egg structure. You can give it to your manuscript or wherever is applicable. You can give this boiled egg and you can discuss as I mentioned. And this is the Excel file I have downloaded earlier. You can delete the unnecessary parameters like that. I am deleting those things. So like that, you can give a table in your manuscript. There will be molecular name, canonical smile. You can keep it or you can delete it. You can delete the formula also. So by this, you can give a table in your manuscript, and whatever parameter you require, you can keep, and that unnecessary parameter you can delete from this Excel file. So this was the EDMAP screening. And this is the boiled egg structure. So if you want to know more about this, just follow these 3 research paper. You can get the idea about those parameters clearly. So this was one compound. Then you can analyze the second compounds in same methods. This is the another compound. So by this structure, this is quercetin. If you want to get the target prediction here from the this window, just in the second icon, it is written as there is a symbol of target. Just click on this target and it will automatically go to the switch target prediction and it will give you the result of target fishing. So from this website also you can get the target prediction result like that. So this was the GetNet profiling. So if anyone having any doubt, you can. Yeah.
Sir, in the boiled egg diagram, how the position of the drug says about its effectiveness, like aspirin is in the periphery of the yellow circle. So, is it
Yes.
good to be in the periphery or in the middle or in the leftmost corner? So, how the
Can you
position?
repeat your question?
Actually, my question is how the position of the drugs in that boiled egg diagram says about the
Hmm.
effectiveness or the properties of that drug.
Okay, this boiled egg only. Okay, I, this boiled egg. Oh, I have to share my screen. Is it visible?
Yes, sir.
Okay. So this boiled egg only give you the idea about the blood-brain barrier permeability and gastrointestinal absorption. This boiled egg will not give you the idea of rest of the parameters. It will only focus in 3 factors. That is, one is it blood-brain barrier permeable or not? Is it having the good gastrointestinal absorption, or is it PGP glycoprotein active or inactive? So from this blood-brain barrier, we can get idea of only 3 parameters. The first parameter is most important. Is it absorbed by our gastrointestinal intestine or not? So our compound should be in the white part of this boiled egg. It should not be outside of the white egg. The first criteria is our compound should be in the white part. Okay, first criteria. It should not be the other area rather than this egg. Like it should not be there, should not be like there, should not be there. Some compounds which is having the low gastrointestinal activity, it will be in outside the egg. So we can't consider those compounds because it will not be absorbed by our gastrointestine. So first criteria, it should be in white part. Second criteria is if we are working on disease related to CNS or related to brain, then it should be in yellow part because it should permeable to blood-brain barrier. So second criteria, if we are working on disease related to brain or CNS, it should be in yellow part. Second criteria. Third criteria, if we are working with this uh, disease related to brain, then our compound should be blue. it should be in yellow part, but it should be blue because, sorry, it should be red, because if it is blue, then it will be efflux out from the yellow part, that means from the blood-brain barrier part. So we are trying to insert our ligand to yellow part, and if it is efflux out from the yellow part to the white part, there is no meaning. So if it is red, it will not be efflux out, but if it is blue, then it will be efflux out. So third criteria is, if you are working with disease related to brain, the compound should be in yellow part and it should be red. Fourth criteria is, if we are working rather than any disease related to brain, then our compound should be in white part. If our compound is in red part, it should be blue, because okay, our compound is permeable to blood-brain barrier, but it will be efflux out by the PGP protein, PGP glycoprotein. So if we are working on rather than any disease of related to brain, then our compound, if it is in yellow part, it should be blue. So there is 4 criteria about this boiled egg to analyze your drug. Clear now?
Yes, sir.
If we are working on drug design against drug-resistant bacteria pathogen, Staphylococcus MRSA, our ligand compound should be blood-brain non-permeable and PGPS. Yes, Mohit Bhattacharjee, your if you are working on any antimicrobial activity by your ligand, if you are working on Staphylococcus MRSA, then your compound should be non-BBB permeable, because you are not necessarily permeable to BBB, because if it permeable to blood-brain barrier, it will interact with the CNS. It may give other side effects. So it should not be blood-brain barrier permeable. And yes, if it is permeable, the PGP glycoprotein should be yes. If it is permeable, it will efflux out from the blood-brain barrier. So your answer you can get from this boil egg also, or from this table also, from this pharmacokinetics table also, you can get your answer. So anyone having any doubt? Any question till now? If anyone having any problem like till now, I can repeat the process again. Can you please repeat about the open value? Open value I have shown again. And okay. So next we will see the bioactivity prediction of the ligand. So what we have seen till now, that we have our ligand, we have retrieved from our databases. If it is not available, we have drawn the structure, we have generated the required format of our structure, we have seen the target, we have seen the toxicity, we have seen the end profile of our ligand of interest. So by target fishing, we can just get the idea of target, which target it can be interact with. But by predicting the bioactivity, we can get the idea about the disease, if it is working on this type of disease or not. So for this, we have to create one account a website known as Where to Drug. This is one website known as Where to Drug. So this is known as PASS. So we predict the bioactivity using the PASS online tool. So using this tool, you have to have one account in your Way to Drug server. So to get this account, just go to the registration part and complete the registration with this registration form. So after that, you can get one login option. So I have my account, so I will log in with my account. Just click on the go to prediction. I will give my username and password. So this is the portal of Pass Online. Here you can predict new compound by clicking predict new compound. I will click this on. Now there is three options to upload your compound. One is Smile, one is Molfy, one is Marvin JS Smile. So this is the easiest way to input our file. It is Smile format. So I will click on Smile. Now here written as insert your Smile here. So I will get my Smile from the PubChem. So this is the Smile. I am copying it. Just copy it and paste. And click on get prediction. Now you can see there is two value. One is PA and another is PI. And third one is activity. And if you scroll down, there is so many results is there. You can see there is so many results. So what is PA and what is PI? PA means probability of activity and PI means probability of inactivity. That means if the PA value is higher than the PI, that means there is the higher chance of showing this activity by this compound. And if the PI value is higher than the PA, that means the probability of not showing this activity is greater than the possibility of showing this activity. Another thing is, if those PA and PS score are similar, like that there is 117 and there is 115. That means there is 50% chance of showing this activity by this compound. Okay. So how we can select the activity? So there is, if PI is less than PA, that means this activity will not be shown by this compound because the probability of inactivity is higher than probability of activity. That means this compound will not show the activity. So we can filter out the result by showing probability of activity should be greater than the probability of inactivity. So there is the first criteria we can filter out by clicking that probability of activity is greater than the PI. That means there should be, the result should be higher in green area, that is probability of activity, and we'll click OK. So these are the results showing that those activity is showing higher than the inactivity. So this is one filter. Next, if you want to more authentic data, you can select that probability of activity should be greater than 0.3. That means that probability of activity should be greater than 30%. The chances of showing the activity by this compound should be greater than 30%. And click OK. It will narrow down the result. Okay, so you can get more accurate result by clicking the probability active at 0.3%. But you have to get the highest confidence score. That means the chances of this activity should be more. Then you chose PA greater than 0.7. That means probability of activity should be more than 70%. So this is the strongly predicted values that this compound is showing 70% is chances there or more chances there, it will show this type of activity. So when I will click the PA greater than 0.7 and click OK, it will show the result. Now see, it is sorted by higher result to lower result, higher point to lower point. The highest point is 0.955 and lowest point is 0.709. That is, this comma is considered as point. Okay, so this is 0.709. If you multiply it 100, you will get the percentage. That means 70.9% probability is there to showing the activity, and 0.303%, that means 3% probability is there not showing the activity. So you can scroll down and find out the activity showing by this compound. So see, there is antipyretic activity showing by the compound. So if you are working with a ligand having the antipyretic activity, you can choose this ligand because it is having the 93.2% probability that this compound will show antipyretic activity. Then if I scroll down, it is antiseptic. That compound can be used as an antiseptic, and the chances of this compound be an antiseptic, there is 90.3% chances. Then this is the NADPH inhibitor, NADH inhibitor, NADP plus inhibitor. If you have to inhibit NADPH, you can choose this compound. If you want to inhibit NADH, you can choose this compound. If you want to inhibit NADPH plus, you can choose this compound. There is glutathione thioesterase inhibitor, glutamine endopeptidase 2 inhibitor. According to your requirement, you can see is this inhibitor is the good choice for your drug or not. So I am going I am scrolling down to show you one thing. So see, this is anti-inflammatory activity. This compound having the anti-inflammatory activity showing 76.2% chances is there. This compound will act as a anti-inflammatory agent. Anti-eczematic, Let me continue with another compound. I have to show one thing. See, I have entered another compound known as nimbolite. So when I entered the compound smile of the compound nimbolite, and I have chose the filter PA greater than 0.7, I get only 1, 2, 3, 4, 5, 6, 7, 8. Only 5 activity I have found. But you can see that this is showing the anti-neoplastic activity. Okay, this compound is showing anti-neoplastic activity. The compound is showing anti-leukemic, that means leukemia. This compound can be used as anti-leukemic activity. The compound is showing as anti-neoplastic activity, that is colorectal cancer. This compound is showing prostate cancer treatment. The compound showing is anti-neoplastic for colon cancer. The compound showing anti-neoplastic for breast cancer. The compound showing beta glucuronidase inhibitor, and the compound showing anti-neoplastic of lung cancer. Okay, so what can you predict from this compound? That this compound will act a very good drug for anti-cancer treatment. But this compound will not be work in any other kind of treatment. Like this compound is not showing any target for diabetes. This compound is not showing any target for cardiovascular disease. This compound is not showing any target for kidney disease. Okay, so what can you get? That this compound is showing the target related to cancer. And one interesting thing that in my PhD, I have worked with the compound nimbolite. This is my this compound I have worked with in my PhD, and I have worked on lung cancer, non-small cell lung cancer, and I have found very good result in vitro studies of very 2 to 3 cell line in lung cancer. I have found very good result by this compound. So in silico studies, I have predicted that nimbolite will act as a very good drug against lung cancer. I have do the ADMET profile, I have do the toxicity profile, I have do the uh, pass activity. By pass activity, you can get that it is showing the anti-lung cancer activity. And I have got for the molecular docking and molecular dynamic simulation, I have gone for the QCR, and I get this compound will very good effect in and non-small cell lung cancer. And as a result, in vitro studies also supported my in silico work, and I found yes, this compound having good activity against lung cancer. So by this, I get an idea by the in silico studies that yes, my compound having the result related to lung cancer, and I have proceeded with this compound for in vitro study. If I found that this compound is not showing any kind of cancer anti-cancer activity, then I may not be going for the in vitro studies with this compound. So like that, you can get the chances for getting the more accurate result in vitro or animal model studies by in silico analysis. So this was the bioactivity prediction with your compounds. Okay, so for to use this server, you just need to get registered. Simply you got registered, and you can use your compounds to see the bioactivity. If we were working with plants or insects, I would design a graph like this. Shown for insect, you can get the idea from this type of drug designing, but for plants, I have no idea because the biochemistry for plants and animals too is totally different. So I don't have any idea what is the biochemical process going on in plants and it is how it is different from animal. So for insect, it will follow this process, but in plants, it may be follow or may not be, because in plants there is no blood-brain barrier. In plants there is no gastrointestinal activity profiling. There is no, so this is totally different from the animal model. So I think it will not be working treat disease for plants. Any question till now? Any doubt? Any question? So, a homework for all of you. Just select one compounds from yourself. Don't download it from the server. Draw it. Generate the smile. Then predict the target with this compound. Predict the toxicity. Predict the ADMET profile. Predict the bioactivity of your compound of interest. This is the homework I have given you. So you can do it, and after performing by yourself, if you face any difficulties, please let me know. I will try to give the solution for the difficulties. Without trying, you can't get the idea if you are facing any problem or not. So you should try at your home after completing the class. And if you are facing any problem, please let me know. And for Sean, I think you are not present. So in your question, if I were working with plants or insect, I would design a drug like this. For insect, you can follow this process, but for plants, you can't follow this process because these algorithms are mainly involved using the human system or animal system. And the biochemistry and the other pathways of plants and animals are totally different. So those algorithms will not work with the plants. But for insects, it may work. So homework is just you select one compound, type this compound in PubChem, and get the idea of this structure. Don't download it from there. You draw this structure using ChemDraw. Then from ChemSketch, get the IUPAC name of this compound, get the name of this compound, then generate the smile of this compound. Then you go for target prediction, then go for toxicity prediction, then go for ADMET prediction, then go for bioactivity, And if you get any difficulties during your this process, please let me know. I will repeat if necessary.
Thank you, Rajat, for your wonderful session. Thank you, all of you, for joining today's session.
Thank you, Shobham.